Well, thank you for attending today's talk. It's a great pleasure to introduce Gregg Semenza. He's received a MD and PhD in his education and had training at a number of illustrious institutions, including Harvard, University of Pennsylvania, Duke, and Johns Hopkins. Currently, he's the C. Michael Armstrong Professor of Genetic Medicine at the Johns Hopkins School of Medicine. He's also the founding director of the vascular program at the Johns Hopkins Institute for Cell Engineering. Gregg Semenza's lab has made the notable discovery of identifying hypoxia-inducible factors and has spent a lot of time identifying their roles in physiologic processes and pathology. More recently, his efforts have been toward finding chemical modifiers of HIFs, and some of those have already reached the clinic where they're in various clinical trials for disease indications. For this exciting work, he has been awarded a large number of prizes I don't have time to go into today. I'll just mention a few, including the Gardner International Award, the Lasker Prize, and in 2019, the Nobel Prize in Physiology and Medicine. We're very lucky to have him today. Unfortunately, he was unable to attend the event live, and so he's recorded his presentation, and therefore we will be unable to take questions after the presentation. Nevertheless, I think you'll be very excited to hear what he has to say, and I'll turn it over to Gregg.
Thank you, I'll try to give you a brief introduction to the role of the hypoxia-inducible factors that we discovered in physiology and in medicine. Oxygen homeostasis is the balancing between the demand for oxygen in every cell of the body, principally for the mitochondria to make energy, and the supply of oxygen, which is, of course, picked up in the lungs by red blood cells that are then carried by the heart and blood vessels to all the cells of the body. And the hypoxia-inducible factors play a critical role in balancing oxygen supply and demand in every one of the 100 trillion cells in your body. I'll give a short introduction to the discovery of HIF1, tell you about its role in cardiovascular disease and cancer, and end by a short story about life at high altitude. When we started this work, we knew that the kidneys made a hormone called erythropoietin. EPO was secreted into the bloodstream, bound to erythroid progenitor cells in the bone marrow, and stimulated their survival, proliferation, and differentiation into red blood cells, which are then released into the circulation and carry oxygen to every cell in the body. The cells in the kidney that make EPO can sense the oxygen concentration and respond to reduced oxygen availability by increasing their production of EPO, thereby increasing red blood cell production and oxygen delivery capacity. So this is a physiological system designed to maintain oxygen delivery to the cells. In chronic kidney disease, the production of EPO by the kidneys is impaired, and patients with chronic kidney disease become anemic. Fortunately, the patients will respond to EPO if it's provided as a recombinant protein. And so the cloning of the EPO gene resulted in this new therapy that was very efficient at maintaining red blood cell production in patients with chronic kidney disease. The main question that we confronted when we started this work was what was the mechanism that controlled EPO production within the kidney. And we were able to identify a short DNA sequence that we call the hypoxia response element. It's located just downstream of the EPO gene. And this 33 base pair sequence has the property that it can be taken out of the EPO gene, inserted into a heterologous reporter gene, and the expression of that gene will be induced by hypoxia or low oxygen conditions. We then identified a protein that bound to the HRE in hypoxic cells, and we called this protein hypoxia-inducible factor 1, or HIF1. We used its binding to the DNA sequence as a way to purify the protein. And we found that it was composed of two subunits, HIF1 alpha and HIF1 beta. These two proteins dimerize, they bind to the HRE, and then they recruit proteins called coactivators that serve as a bridge to the transcription initiation complex containing RNA polymerase II that transcribes the EPO gene into RNA. We generated antibodies against HIF1 alpha and HIF1 beta, and we found that the HIF1 alpha levels dramatically increase at oxygen concentrations below 6%. Most cells in the body are exposed to between 2 and 6% oxygen, so you can appreciate that any decrease in oxygen availability in vivo would occur along the steep portion of this curve, providing a mechanism for a graded transcriptional response to hypoxia. The more severe the hypoxia, the greater the production of HIF1 alpha and the greater the expression of genes such as EPO. The basis for this oxygen-regulated expression of HIF1 alpha was discovered in 2001 when several labs found three proteins called PHDs. This stands for prolyl hydroxylase domain proteins. And these proteins use oxygen and alpha-ketoglutarate, a small molecule metabolite of glucose, to hydroxylate HIF1 alpha. So they insert an oxygen atom into one of the proline residues in HIF1 alpha. The other oxygen atom is used to split alpha-ketoglutarate into CO2 and succinate. When the proline residue is hydroxylated, it enables the binding of a protein called VHL, the von Hippel-Lindau protein. And when VHL binds to HIF1 alpha, it triggers the degradation of the protein. So this reaction occurs under normoxic conditions when oxygen is available. Under hypoxic conditions, the reaction is inhibited because of the lack of oxygen, and the HIF1 alpha protein can rapidly accumulate within cells. In addition to this proline hydroxylation, there's an asparagine residue in the transactivation domain of the protein that's also hydroxylated by another protein called FIH1. This protein uses the same biochemistry, and in this case, the hydroxylation prevents the binding of the coactivator proteins that I showed you in the previous slide. Again, under hypoxic conditions, these proteins can bind to HIF1 alpha, and the protein is competent to activate the transcription of the EPO gene. So you can see that these oxygen-dependent modifications control both the half-life of the protein and its specific activity as a transcription factor. And this provides a mechanism to transduce changes in oxygen availability to the nucleus as changes in HIF transcriptional activity. So hypoxia can occur either to an increase in oxygen demand, so if there are increased numbers of cells consuming oxygen, or a decrease in oxygen supply, as occurs in the case of vascular disease, where blood flow to a tissue is impaired. In either case, HIF1 is induced, and then there are, besides EPO, there are over 4,000 genes to which HIF1 can bind and activate transcription in different cell types. Some of these genes serve to increase oxygen delivery, like EPO, or vascular endothelial growth factor, VEGF, which stimulates blood vessel formation. And some of these genes allow the cells to survive under low oxygen conditions. And the classic metabolic adaptation is the switch from oxidative to glycolytic metabolism as a way of generating energy. And all of the glycolytic enzymes are activated transcriptionally by HIF1. We knocked out the HIF1 alpha gene in mice, and we found that mice that were homozygous for the knockout allele, so they did not express any HIF1 alpha, arrested in their development in mid-gestation, And these embryos had major defects in the heart, the blood vessels, and in the production of red blood cells. So all three components of the circulatory system are dependent on HIF-1 alpha for their normal development. Now, animals that have one wild type copy and one mutant copy develop normally. And as you'll see, they have been useful for us in evaluating the role of HIF-1 in various disease models. Well, the story became a little more complicated with the discovery that HIF-1 alpha has two siblings, HIF-2 alpha and HIF-3 alpha. All three of these proteins are oxygen regulated and dimerized with HIF-1 beta, but whereas HIF-1 alpha is expressed in all nucleated cells of virtually all metazoan species, HIF-2 and HIF-3 are only expressed in certain cell types and only in vertebrate species. So these are evolutionary add-ons that allow the developmental programs that are critical for developing the cardiovascular and respiratory systems. Well, what's the evidence that the HIF pathway is critical for red blood cell production in humans? There's a human condition called hereditary erythrocytosis in which affected individuals make too many red blood cells. And the first individuals who were identified with this disorder were found to have a mutation in the gene for the EPO receptor that allows this receptor to be active even if EPO has not bound to it. And as a result, this stimulation of the EPO receptor results in increased red blood cell production. The other individuals with this condition who have been identified either have defects in the prolyl hydroxylase that hydroxylates the HIFs, or in VHL, the protein that binds to the hydroxylated HIFs. So these mutations either reduce the ability of the hydroxylase to modify HIF-1 or reduce the ability of VHL to bind to the hydroxylated protein. And these mutations prevent the degradation of the HIFs, and that results in increased EPO production. And then finally, individuals were identified who have mutations that change a single amino acid in the HIF-2 alpha gene, which is called EPAS1. And these mutations inhibit the ability of the hydroxylase to modify just HIF-2 alpha. And it turns out that just turning on HIF-2 alpha is sufficient to increase red blood cell production. So you can see that these mutations in this pathway all lead to increased red blood cell production and demonstrate the critical role of this pathway in controlling erythropoiesis in humans. Now, I mentioned to you that the PhD enzymes, besides using oxygen, use alpha-ketoglutarate as a substrate. And when this was discovered, it made it immediately apparent that one could use an analog of alpha-ketoglutarate, such as this compound that's called dimethyloxylglycine, or DMOG, to inhibit the hydroxylase activity. And then that inhibition of hydroxylase activity would result in more HIF-1 alpha and HIF-2 alpha and more EPO production. And 4 different pharmaceutical and biotech companies have developed drugs that inhibit the hydroxylases. And you can see these drugs share the backbone of alpha-ketoglutarate and DMOG, and then they have additional residues that give them drug-like properties. And so these 4 drugs are all in phase 3 clinical trials for the treatment of anemia in patients with chronic kidney disease. So whereas recombinant human EPO requires injections, these drugs, of course, are small molecules, and so they can be given by mouth as pills, and therefore are much more convenient for the treatment of anemia in patients with chronic kidney disease. And the first of these drugs to be approved was roxadustat, based on two large clinical trials in cancer, in China. One group involved patients with chronic kidney disease who did not yet require dialysis, and the other study was in patients with chronic kidney disease who were already on long-term dialysis. And in both groups, the treatment with roxadustat was very effective in stimulating red blood cell production and correcting the anemia. So this drug has been approved for use in China, Japan now, and several other countries. This may change the management of patients with anemia, these drugs may be used in addition for, as you'll see, for other conditions where one would like to increase HIF-1 activity. One of those diseases is ischemic cardiovascular disease. And this is the leading cause of death in certainly in Western countries. And it comes in two types: coronary artery disease, where there's a narrowing in one of the major coronary arteries supplying blood to the heart. This results in chest pain. And then if the artery becomes completely obstructed, can result in a heart attack. The large blood vessels supplying blood to the legs can also become obstructed, and this results in leg pain. And if it's not possible to relieve the obstruction, this will eventually require amputation of the limb. And you can see that this patient is getting very little blood to the lower leg. All the blood that's going down into the lower leg is coming off of these collateral blood vessels that branch off of the major artery before the obstruction. And so this patient is very likely to have ischemic pain even when not walking, so just at rest. The lack of blood flow will result in ischemic ulcers that do not heal. This can lead to infection of the tissue, and as I said, may eventually require limb amputation, because there's no effective therapy if it's not possible to open up the closed artery. This condition is seen primarily in older individuals, and so we wanted to model the effect of aging in mice. And in this experiment, we ligated or cut off the blood supply to the femoral artery, which supplies blood to the lower leg. And we were able to image blood flow in the leg non-invasively with a technique called laser Doppler perfusion imaging. And you can see that before the therapy, there's good blood flow. Before the treatment, there's good blood flow in both legs. After the surgery, the blood flow in the leg here is dramatically reduced, and we could express this as the ratio of blood flow in the instrumented to the non-instrumented limb, and it starts at 1, then drops below 20%. And then in young, healthy mice, there's a gradual recovery of blood flow over the course of 5 weeks. If we do this experiment with an 8-month-old animal, the recovery of the blood flow is not as effective. And if we use a 20-month-old mouse, remember mice live 2 to 3 years, we see very little recovery of blood flow at all. Then at each age, you can see that animals that are heterozygous for the HIF-1 alpha knockout gene, so they have one normal gene and one HIF-1 alpha knockout gene, these heterozygous mice at every age recover less well than the wild type littermates. So you can appreciate that just by changing the genotype at one locus and the age of the mice, we're able to generate a range of outcomes from very efficient recovery of blood flow to very little recovery of blood flow. And this has important consequences for the maintenance of tissue viability. So in the young wild type animals, 100% of the mice recover without any permanent tissue damage, what we call limb salvage. But if we look at 8-month-old animals, 40% of these mice either had soft tissue necrosis in the leg or a below the ankle amputation of one or more toes. And then in the old mice, 15% of these animals developed an above the ankle amputation where they lost the entire foot because of the failure to recover blood flow adequately. And then you can see that at every age, the heterozygous mice have a more severe phenotype than the wild type littermates. At the molecular level, we could look at the induction of HIF-1 alpha protein in the ischemic limb of the 2-month-old mice. So this, we measured HIF-1 alpha protein 3 days after the femoral artery ligation. And you can see that in the ligated limb, HIF-1 alpha is induced, whereas in the non-ischemic limb, there's very little HIF-1 alpha. In the 8-month-old mice, ischemia induced HIF-1 alpha, but to a lesser extent. And then in the 20-month-old mice, there's very little induction of HIF-1 alpha at all. And in the wild-type mice, the HIF-1 alpha induction was also impaired. So with aging, there's a deficiency in the production of HIF-1 alpha under ischemic conditions. We engineered an adenovirus that encoded a form of HIF-1 alpha where we had made a deletion and several point mutations that render this protein resistant to oxygen-dependent degradation. So this protein will be expressed regardless of the oxygen concentration. And so we injected this constitutively active form of HIF-1 alpha in the form of an adenovirus into mice at the time of femoral artery ligation. So we injected the virus into the muscle of the ischemic limb and then looked at the recovery of blood flow in animals that received the HIF-1 alpha adenovirus as compared to a control adenovirus encoding bacterial beta-galactosidase. And you can see that the control animals recovered about 55% of their blood flow by 3 weeks, whereas the animals that received the HIF-1 alpha adenovirus had over 80% recovery of blood flow in the same period. Now, these were young, healthy mice, and as I mentioned, those mice actually will recover fine on their own. So we repeated the experiment in 8-month-old mice, and now you see that the control animals only recover about 35% of their blood flow. And injection of the HIF-1 alpha adenovirus brings that up around 50%. So you can appreciate that the treated older animals have a recovery that's similar to the control younger animals. So we've been able to correct the age-related impairment of vascularization in response to anemia through the injection of this adenovirus encoding constitutively active HIF-1 alpha. And the reason for the effectiveness of the HIF-1 alpha adenovirus is that HIF-1 turns on the expression of a whole series of genes that encode secreted proteins that bind to cognate receptors that are present on endothelial cells, endothelial progenitor cells, bone marrow-derived cells that are recruited to the ischemic tissue and promote the tissue vascularization. And so it's this coordinated response that HIF-1 alpha orchestrates that allows the recovery of blood flow in these mouse models of ischemic disease. Now, cancer is the opposite situation in which the cancer activates the HIF pathway. And this is good for the cancer, but it's not good for the patient. And I'll start the story about cancer with this distinguished gentleman, Professor Eugen von Hippel, who was an ophthalmologist working in Heidelberg. at the turn of the 20th century. And what Professor von Hippel noticed was that he found patients who had these tumors of the retina. And he wrote a paper, he called this "About a Very Rare Disease of the Retina." And he noticed that these retinal tumors, these retinal hemangioblastomas, were found to run in families, suggesting that they were due to an inherited genetic defect. And he published this in 1904. In 1926, Lindau reported that patients with this disease often also had kidney cancer. And so this syndrome of these retinal hemangioblastomas and renal cell carcinoma became known as the von Hippel-Lindau syndrome. And patients with this disease have a mutation in the VHL gene that I told you about earlier. As a result of this mutation in VHL, the protein cannot bind to HIF-1 alpha and HIF-2 alpha. And as a result, at high oxygen concentrations, HIF-1 alpha and HIF-2 alpha are not degraded. So you can see that there's as much HIF-1 alpha and HIF-2 alpha in these cells at 20% oxygen as there is at 1%. If we reintroduce a functioning copy of the VHL gene into the cells, now you see the HIF-1 alpha and the HIF-2 alpha are degraded under hypoxic conditions. This is an example of one of these cancers that are found in VHL patients. So the patients have both retinal hemangioblastomas, cerebellar hemangioblastomas, and clear cell renal carcinomas. And they're called clear cell because the cytoplasm does not stain well with H&E staining and gives this clear appearance. But you can see that there's very intense staining of every nucleus. And that's because this section has been stained with an antibody against HIF-1 alpha. So the tumor is making very high levels of HIF-1 alpha and HIF-2 alpha. And this is critical for tumor formation. And what's known is that patients with the von Hippel-Lindau syndrome have a mutation in one copy of the VHL gene in their germline, and then the second mutation occurs in a somatic cell in the kidney. And when both copies of the gene are lost, kidney cancer results. And the same thing happens in the eye, the same thing happens in the cerebellum to form hemangioblastomas there. And what's known is that mutations that have a more severe effect, that is to say they interfere with the binding of VHL to HIF-1 alpha and HIF-2 alpha to a greater extent, those patients are more likely to develop renal cell carcinoma. And renal cell carcinoma, once it spreads from the kidney, becomes metastatic, has a very grim prognosis, with less than 12% of patients surviving 5 years. And that's because there's no effective therapy for metastatic renal cell carcinoma. And so it was quite remarkable when a biotech company, Peloton, developed a small molecule inhibitor of hypoxia-inducible factor 2. This small molecule binds to HIF-2 alpha and prevents it from interacting with HIF-1 beta. So if the heterodimer does not form, the protein cannot bind to DNA and cannot activate transcription. So this compound selectively inhibits HIF-2 because it cannot bind to HIF-1 alpha. And so this drug was tested in 50 patients with advanced renal cell carcinoma in a phase 1 trial, which is a trial that's just done to be sure that the drug is safe. And it was given to patients who had failed every existing therapy. And what was found was that among these 50 patients, one of the patients had a complete response, meaning the tumor was completely eradicated. Several patients, you can see here, had a partial response where the size of the tumor decreased by at least 30%. And then there was a large group of patients who had stable disease, meaning that the tumor size did not increase more than 20%. And so this is a remarkable finding for a phase 1 trial. And so this drug is now in advanced trials, both for kidney cancer and also for solid cancers. And this provides proof of principle that inhibiting HIF activity has anti-cancer effects. Well, I'd like to end by telling you a little bit of what we've learned about the evolutionary adaptation to life at high altitude, specifically on the Tibetan Plateau at altitudes of 3,000 to 4,500 meters. And people have lived on the Tibetan Plateau for over 20,000 years. No, they don't live here, but they do live here. And what's remarkable is that if you or I went to Tibet, as I did in 2017, our red blood cell counts will increase because there will be much less oxygen getting to our cells. Lhasa, the capital of Tibet, is at an altitude of 3,650 meters, and the oxygen level in the air is 14%. So that's only 2/3 of what we breathe at sea level, 21%. And I took this picture because to illustrate that this family, these young children, have lived their entire lives being exposed to this reduced oxygen level. And what's remarkable is that the people living in Tibet don't increase their red blood cell counts in response to the hypoxia. Because, increasing your red blood cell count is fine if you've had blood loss, but at altitude, the problem is not too few red cells, it's too little oxygen. And when the red cell count gets high in the blood, it can have adverse consequences. And in fact, patients who have chronic mountain sickness, these are people who live in Tibet who are not able to adapt. And these individuals have high red blood cell counts, they have pulmonary hypertension, and they also have cerebral edema. And these patients will die if they are not brought down from high altitude. So most of the people who live in Tibet have adaptations in their genome that have been selected over time that allow them to live normally despite this reduced oxygen availability. And so with the development of next generation high throughput DNA sequencing, it became possible to study single nucleotide polymorphisms. These are differences in the DNA sequence that are scattered throughout the genome, and to ask, are there differences that are present in Tibetans, but are not present in the ethnic group that they probably arose from originally, which is the Han Chinese. So in this study that was carried out by Cynthia Beale's lab, and this is a picture I took of Cynthia in front of the Potala Palace in Lhasa 3 years ago, what Cynthia's lab did was to compare 35 high-altitude Tibetans and 84 Han Chinese DNA sequences. And they looked at over 500,000 single nucleotide polymorphisms. And then for each one of those, they compared the frequency of the two different possible alleles and asked, were they significantly different in the two groups? And the test of significance, because there were over 500,000 comparisons that were made, the p-value had to be very small. And there were only 8 single nucleotide polymorphisms that were significant in this analysis, and all 8 were at the locus that codes for HIF2 alpha, the EPAS1 gene. And what's more, the Tibetans who had these polymorphisms had lower hemoglobin levels than those who did not have these polymorphisms, suggesting that these polymorphisms allowed this adaptation, which was the maintenance of a normal hemoglobin level. Well, further sequencing was done and more polymorphisms were found in the EPAS gene, and 5 polymorphisms that were found clustered together were present in Tibetans and completely absent in Han Chinese genomes. Well, they weren't just absent in Han Chinese genomes, they were absent in the genome of every other ethnic group on Earth. And the only other DNA sequence where these polymorphisms were found was in Denisovan DNA. And you may ask, what is Denisovan DNA? Well, Denisovan DNA is DNA from a Denisovan. And the Denisovans are a hominid species that was a evolutionary dead end that, like the Neanderthals, diverged from humans between 500,000 and 1 million years ago. And whereas the Neanderthals populated Europe, the Denisovans populated Asia, and particularly in Melanesia, where Denisovan DNA is still present in the genome of present-day Melanesians, accounting for 3 to 5% of the total human genome sequence. And so the idea that DNA that's retained is likely to be valuable, to have some beneficial effect. And so it was remarkable, and the way the Denisovans were identified was that in a cave in Denisova, which is located in Siberia, a pinky bone and a very large molar were identified. And DNA was extracted from that pinky bone and sequenced and was found to be unique. It was not Neanderthal, it was not human, and so this was called Denisovan. Well, this cave in Siberia is a long way from Tibet, and Siberia, this is not at elevation either. So the question is, why would Denisovans have a DNA sequence that seemed to be protective at high altitude? And so the obvious question was, did any of those Denisovans get to Tibet? And so the story became very interesting last year with the following very interesting results from a cave in China. So this is a cave, the Baixia Karst cave in Shahe, China, which is an altitude of 3,200 meters on the edge of the Tibetan plateau. 40 years ago, a monk went into this cave to meditate and pray, and he found something very interesting, a jawbone with 2 very large teeth. And he thought that this was sufficiently interesting that he presented it as a gift to the 6th Gongfang living Buddha, who, being a wise man, gave it to the anthropology department at Lanzhou University, where it sat on a shelf for several decades until an anthropologist visiting from Germany was shown the fossil in 2019. He saw the large teeth and immediately said, this must be Denisovan. And he was able to extract protein from the jaw, analyze that protein, and show that it had a sequence that was found in the Denisovan DNA that had been isolated from the pinky bone in Denisova. And furthermore, dating of the fossil indicated that it was 160,000 years old. So Denisovans were present on the Tibetan plateau 160,000 years ago. And by about 40,000 years ago, that denisovan sequence at the HIF2 alpha gene had become selected for and was present in the genome of many Tibetans. So just to summarize what I've told you, the HIF pathway plays a critical role in development, physiology, medicine, and even in evolution. And the main focus now, of course, is to see how we can apply what we've learned to the treatment of common human diseases such as cancer and cardiovascular disease. Thank you very much for your attention.
Well, thank you for that wonderful presentation. I think that it's a great illustration of how basic science can go all the way to discoveries that lead to medical treatments, that lead to understanding our evolutionary history, and reach into all kinds of areas that I think no one probably would have expected when the original research was done. So that was very exciting. And, you know, HIF1 continues to be of interest in a wide range of tissues and diseases. It's also very interested in my field of aging, where, as I illustrated in the questions, both activation and inhibition of HIF1 can be beneficial for aging in different contexts. So let me just again thank Dr. Semenza for the presentation. I'm sorry we can't take questions, and I encourage you to stay on for the rest of the conference. Thanks a lot.